So today we are going to be learning about Punnett squares, what they are, and how we use them. So the first couple slides I'm going to go over, you need to copy those down in your journal. Then the last 3 slides are going to be practice examples we go over together. So first page of notes. A Punnett square is a tool to predict the probability of certain traits in offspring that shows the different ways alleles can combine. So if we know the parents and we know can figure out their genotypes, we can use a Punnett square to try and predict what will happen with their offspring. So the Punnett square shows all possible combinations of genotypes when that result when a gene is crossed. So it just shows you what could happen and the probability of each one of those outcomes. In your Punnett square, you'll see we'll have basically a Punnett square is 4 squares put together. Each of those 4 squares represents a 1 in 4 probability or a 25% chance that a certain genotype will be inherited. So this here we have the steps for doing a Punnett square. We're going to go over these. I'm going to be writing on this slide as I talk, and then you also need to copy this down in your notes. So the first step is to draw the Punnett square. I already have one drawn here for you. So on the right, you see those 4 boxes. If you don't have one pre-printed on a worksheet or something like that for you, then you need to draw it out. The next thing you're going to do is fill in the parents' genotypes. You will put the male's genotypes on the top and the female's genotypes on the left. So we're going to use an example of 2 parents that are both, they're both going to be heterozygous. Remember, heterozygous means they have one dominant allele and one recessive allele. Sometimes when you work out these problems, they'll tell you what letter to use. If it doesn't tell you what letter to use, you can pick your own. Try and pick something that looks different when it's capital and lowercase. So things like O and X and C aren't good examples. Things that you want things that look different. So A, B would be a good example. We're going to use B. So the male's genotype goes on the top. We said they're both heterozygous. So I'm going to draw, let me turn on that mode. I'm going to draw up here the male's genotype. So the male is capital B, lowercase b. You'll see I put one letter above each column. And I also said that the female was heterozygous. So then over here on the left, I'm going to put capital B and lowercase b. I'm going to try to use different colors so it's a little more clear when we get down to step 3 and 4 exactly what I'm doing. Okay, so then step 3 is to distribute the males. All right, accidentally deleted the ink there, so pause the video, got it back to where it was. So as I was saying, step 3 is to distribute the male's alleles. So you're going to go over here, remember the male is on the top, the red part where I've drawn on the top, and I'm going to distribute those letters. So in this column, I'm going to take this capital B, and I'm going to put it in this square. And then I'm also going to distribute it down below, so this square down here. So you have a capital B down here. And then in the next column, I'm going to look at the lowercase b. I'm going to distribute that lowercase b through the 2 squares below that. So I'm going to draw a lowercase b there, and then also a lowercase b there. And that is step 3. Step 4 is to distribute the females' alleles. So I'm going to take the letters on the left and put them in each of the rows that they are in. So this capital B, to the right of it, there's 2 boxes. I'm going to put it into each of those. So I'm going to draw the capital B here. And you'll see that I'm keeping the colors consistent so you can see where they came from. And then also in this box here. Okay. And then I'm going to come down to the next row and I'm going to look at the lowercase b, and I'm going to distribute the lowercase b into this box and then also to the one next to it. And so this now shows us all the possible genotypes that can be produced by these 2 parents. And then we can also use that information to figure out the phenotypes. So step 5 is to determine the probability of each genotype. So the first thing I always do is I list out all the possible genotypes that I see inside the Punnett square. So in the top left corner, I see a capital B. I'm going to change to it, so I'm going to change to a different color. I see a capital B and a capital B. And then in the top right and the bottom left corners, I see a capital B and lowercase b. So I'm going to write capital B and lowercase b. Even though I see it twice, I'm just going to write that once. And then the other thing I see in the bottom right is a lowercase b and lowercase b. Right. So now that I've listed out the possible genotypes, I need to figure out the probability of each. So remember in the previous slide, I said that each of the 4 boxes represents a 1 in 4 chance or 25% chance I'll have that genotype. So we're going to start with a capital B, and I'm going to look up here and I see that in only one of the 4 boxes do I see that genotype. So that means it has a 1 in 4 chance, which is also equal to 25. Whoops. Let me erase that and try again. 25% chance on the homozygous comment, or capital B, capital B. Okay, I'm going to do the same thing for a heterozygous. I'm going to look and I see that it's in 2 different squares, in the top right and the bottom left. So there's a 2 in 4 chance that it can be heterozygous, which is equal to 50%. Okay. And then finally, the last one, lowercase b, lowercase b. We are going to see how many squares that's in. We see that it's only in the bottom right. So that means it's a 1 in 4 chance, which is equal to 25%. So one of the ways you can check to see if your answers are correct is if you add up all the fractions we see here, it should always add up to 4 or 4 over 4. And then if we add up all the percentages, it should always add up to 100%. I do want to point out for heterozygous where I put 2/4, you could have also put 1/2 there as well. All right, and so then for the step 5, we need to determine the probability, step 6, determine the probability of each phenotype. And so I'm going to take these genotypes and turn them into phenotypes. We're going to say that we're talking about blonde hair and brown hair. So we'll say that capital B represents brown hair. So I'm going to write that, let's write that over here. B equals brown and lowercase b equals blonde. Okay, and we're working with complete dominance here. So if we see a genotype that has 1 dominant allele or 2 dominant alleles, then it will have the dominant trait. So over here, we're going to write brown. Okay, and I'm going to look at the information I have here. As I said, this is complete dominance. So if there's at least 1 allele that is dominant in that genotype, then it will have that genotype. So we see that 3 of the boxes have at least 1 capital letter in them. The top 2 boxes and then the bottom left. So there's a 3 in 4 chance that the first one have brown hair, which is equal to 75%. And when we look at blonde hair, in order to have blonde hair, the person has to have 2 recessive alleles. And so we're looking for boxes that have 2 lowercase letters in them. We see that there's only 1 in the bottom right corner, right here. And so that is a 1 in 4 chance, which is equal to 25%. So again, if I add up these numbers, I should have 4 out of 4 for the fractions and 100% for the percentages. Okay, so make sure that all this information is written in your journal. We are going to move on to some examples.